Steve Mitchell, again looking at one science and faith issue. As a Christian, I believe that the God of the Bible really is the creator of the whole universe, and he chose to reveal himself to humans. The Bible is his word, his inerrant word, but we have to be careful how we interpret it. Now, Noah's flood was a real event, but was it a global catastrophic event? Young Earth creationists certainly believe so. They believe that most of the rocks around the world were formed during that event about 4,350 years ago. Now, we need to look at what some young Earth creationists believe is the premier evidence that rocks and fossils were just a few thousand years old. So with that, I'm going to PowerPoint. Welcome to part 3 of Dinosaurs and Noah's Flood. We've looked at evidence that dinosaur eggs and tracks formed over a period of time that just don't fit as deposition during a global catastrophic flood. Too many events and too little time, as my friend Kevin Nelson says. But wait, maybe there's another side of the story. This session is going to look at that. In part 1, we looked at dinosaur eggs and how some nests or clutches show multiple phases of development and are just not possible in a young Earth creation model. In part 2, we looked at dinosaur tracks that are found with tree roots that grew in place and mud cracks that formed as the sediment dried, and they don't fit as part of a global catastrophic flood. But I can just hear some of my young Earth creationist friends saying, yeah, but what about the fact that soft tissue has been found in dinosaur bones? They simply can't be millions of years old. Is that right? Well, let's look. Now, young Earth creation models propose that dinosaurs lived a few thousand years ago, possibly even making it onto the ark. Maybe they just couldn't survive the ice age that supposedly occurred at the end after the flood. Now, geologists recognize ice ages, multiple ice ages, with the last one ending about 10,000 years ago. Scientists have certainly found preserved microscopic remnants of tissue from dinosaurs that's buried inside dinosaur bones. Young Earth creationists would say that this is 4,350 years old. Now, 100 million years would certainly be longer than that. Is it reasonable to propose that soft tissue could be preserved for millions of years? Now, I'm not a chemist, and it's been many years since I had a chemistry course, but even so, I find the studies on this topic really interesting. But I'm going to start with an analogy. Let's think about a fresh peach. Wonderful to eat, especially if it's ripened on the tree. It also makes a great peach cobbler. How long can you expect a ripe peach to stay good? Now, I asked Google, because that's the source of all kinds of wonderful knowledge, and they reported it maybe 3 to 5 days. Now, if it's true, and you're given a peach to eat that's been out for a few weeks, then it's probably not a good idea to eat it. It probably wouldn't look like the peaches in this picture. It really won't be long before the seed will be all that remains looking like a peach. So now let's take a few chunks of this and let's bury them underground, put them in a hole in the ground. How long do you think that will last? Probably not even the 3 to 5 days, right? I would guess that if you buried them and you dug around in a few months, you wouldn't find much evidence that a peach had been there. If you just had chunks of a peach, there's not going to be much evidence. It's going to be hard to find exactly where it was. And I think many people would agree that it's essentially impossible for pieces of peach to remain good when buried for 6 months. However, what if you were to learn that the conditions that the peach slices were buried under were different? Do you think that could make a difference? What if you learned that before burial, the peach slices had been placed in a sanitized jar, covered with sugar water, and then sealed with a sanitized lid? That is, that they had been canned. Even buried, I think you'd agree that they could last for a few months. What if that sealed jar had been sealed with wax or plastic to ensure that the lid wouldn't rust? Online, the report is that they will store for 12 to 18 months at best quality. and they're often safe to eat even after that. I would guess that there would be some material left over from the peach for many years. You see that if you want to know how long an organic material will last after burial, it's pretty important to know the details about what happened to the material after death. It makes a huge difference. Now, I've read that peaches can sometimes be edible 5 years after canning if carefully sealed. Now, that would be 365 times the time that it would be for peaches if they were just set out on a counter. Under special isolated conditions, they can be good for a long period of time. That sealing canning jar effectively isolates the fruit from the environment around it. It sequesters that fruit. That sequestered fruit does not decay at the rate of normal fruit. Now, what does this have to do with the soft tissue of dinosaur bones? Maybe for us to say how long ago the microscopic fragments of material formed, we would have to understand exactly what was the process that sequestered it or isolated the material and all of the conditions that it was under since that time. Now, as for this controversy, personally, I blame the movie of Jurassic Park. I blame it in part for some of the understandings because whenever the movie came out, scientists were asked if it would be possible to find dinosaur DNA and to replicate it like take place in the movie. You remember that they found mosquito blood from mosquitoes that were trapped in amber and tree resin, and they were able to replicate and come up with a T-Rex. Now, scientists, of course, said, no, we can't do that. That's much too long after that. They had to reply this way, and they didn't think this would be something that was possible. And it was a genuine surprise when Mary Schweitzer announced the discovery of blood vessels and cells from a T-Rex fossil. How appropriate is that? A lot of people did not believe it at all. Blood vessels from dinosaurs, really? I mean, you look at these red dots in the pictures that she published. What are those? Were they T-Rex blood cells? Now, that 2005 report by her was just the tip of the iceberg. A lot more has been found today. Now, here's some of the things that have been found. You find bone cells, osteocytes, chondrocytes, or cartilage cells, blood vessels, nerve fibers, nerves, collagen in bone matrix, extracellular sheets of type 1 collagen that form in the non-mineral part of a bone matrix. Find blood cells? Perhaps. It's still a little bit controversial. Some don't accept that. Possibly even DNA fragments. Is that possible? Young Earth creationists often jump on the discovery of soft tissue. They claim it proves that the Earth was young. Their logic is simple. Soft tissue can't be preserved for millions of years. We find soft tissue in rocks, conventionally dated as millions of years old. Therefore, the rocks cannot be millions of years old. And that's consistent with the idea that most of the rocks have been deposited during Noah's flood. Well, let's take a look at some of the discoveries and where they fall in the different age models. We used this chart before. It shows the young Earth line on the left, where you've got up to around 6,000 years ago when the creation would have taken place. It expands out the flood and shows the interpretations of Dr. Tim Clary with ICR, and moving my picture out of the way, Andrew Snelling with Answers in Genesis. What's important is where the dinosaur fossils are found and where evidence of soft tissue is found is all from the Mesozoic, and it's all interpreted by both Clary and Snelling and virtually all other young Earth creationists to have been deposited by the flood. Soft tissue from fishes has been found in the tertiary up above the flood interval according to Andrew Snelling's model, but still in the flood model according to Tim Clary. There's even some amphibians that took place before the age of the dinosaur out of the Permian period, and there may be discoveries later. The consensus ages, but some of them at over 100 million years old. Wow, that's a lot different than the 4,350 years that young Earth creationists would say. So let's take a look at some of the discoveries. Let's consider some of the findings of Mark Armitage from the Dinosaur Soft Tissue Research Institute. This is a young Earth creationist organization that's devoted to studying fossil soft tissue. Its leading investigator is Dr. Mark Armitage, and he's an expert in microscopy. His skill in microscopy is recognized, and he's published in some of his results in peer-reviewed journals. And these are some examples of some of his discoveries of bone cells and tissues from a triceratops. And that's not all. Here's some examples of nerve tissue from the triceratops that Armitage has published. Again, the preservation in these is really good. And how do you go about preserving this? At least it preserves the form of this in great detail. Whether it's the actual material or in its original form is more debatable that way. How could you preserve nerve tissue from a dinosaur that lived over 100 million years ago? How could that happen? So the question then is, how long can bones last? It's not like there's just a simple answer to the question, just like for the peaches that I talked about. You need to know the conditions. As I say, well, it kind of depends. Was the bone buried quickly? Were there any conditions there to inhibit decay? Now, in a humid, warm climate, bones might be completely broken down. In a dry climate, particularly if it's cold, they might last a few hundred years, but still soft tissue should already be gone. Now, if the young Earth creation model were true, and the bones lasted 4,350 years ago, how did that happen? Even for that length of time, normal decay trends just wouldn't preserve soft tissue. They also need to appeal to some mechanism beyond just normal decay. Something has to sequester these cells in a special way. And some scientists have worked very hard on this. Scientists love a new field of questions, and that's what these discoveries opened up, a new place to study. I'd recommend a book by Fazell Rana of Reasons to Believe. It's called Dinosaur Blood and the Age of the Earth. And he lists a whole bunch of things which come together on this. And it's a little book. It's in kind of relatively non-technical terms. And he talks about some of the factors that we know about or knew about in 2016 when this book was written, such as during fossilization, mineral-rich water comes and infuses the remains of the organism. Burial conditions have to be important. Dry and hydrous conditions are also necessary to preserve soft tissues. Sequestering soft tissues from oxygen is important. Soft tissues have to be kept away from environmental enzymes and microorganisms. Iron may help stabilize soft tissues from chemical destruction. Temperature is important. pH can contribute to soft tissue preservation, and minerals can play a key role. A lot of things are coming to play. But then a lot has been learned since 2016. Here's a slide by Mary Schweitzer. This group is recognizing how important iron is in preserving soft tissue back in 2013. Let's fast forward a little bit. Here's a paper that I would really strongly encourage you to look at. It's by a paleontologist whose name is Philip Center, and I'm going to really base quite a bit on it. The title of it is Soft Tissues in Fossil Bone. So it was written in 2022. That makes it newer, fairly current that way. As I'm reading it, would be 1 year old. So this is a lot newer type of information. It incorporates some of the discoveries that have been made since Dr. Rana was writing his book. Dr. Center confirms many of the factors that Fuzz Rana mentioned. He particularly highlights the importance of what are known as Fenton reactions. But I like his examples of 3 different bone scenarios to help us kind of understand that. These are only 3 of many possible scenarios, but I think they help to understand what happened to dinosaur bones. How could they be preserved, or would they be preserved? So we're going to look at our first bone. This is our dinosaur bone A. So it's surrounded by soil that's acidic. It contains calcium, aluminum phosphate. It's porous, it's light, and much of the bone mineral dissolves away. As the bone mineral dissolves, there are cracks that appear. Cracks in bone A tend to be exploited and enlarged by bone-consuming bacteria, by fungi and plant roots, and that invasion of the fungi with the roots tends to enlarge those cracks even more. And this bone, like the bones of most animals that die, eventually is gone without a trace. No fossils, no hard material, no soft material. It's just gone. But that's not the only possibility. We have evidence of ancient bones, so there must be something else working. Now, this bone was laid down in a different soil, an alkaline soil with calcium carbonate, and the climate was cold and it was sandy soil, and such a bone might last much longer. Center tells us that some changes have taken place, though. There's still cracks. After 1,000 years, its blood vessels and osteocytes have succumbed to decay, leaving behind relatively empty blood vessels, but the matrix of the bone is still intact. Over the next tens of thousands of years, as groundwater percolates through the blood vessels, when with it there's fluoride from the groundwater that converts the bone material into francolite and afterward into fluoroapatite, stabilizing it and enabling it to last longer. Permineralization occurs as ions in the groundwater precipitate as mineral crystals form on the surface of the vessels, and they fill the voids and further contribute to the preservation, preventing the entry of destructive agents later. Subsequent drying during freeze-thaw cycles and during wet-dry cycles, or the result of local environments becoming dry, it generates cracks, which create more surfaces on which the minerals precipitate, further contributing to long-term preservation. Such a bone will last for millions of years after its blood vessels and their contents have vanished. We have a fossil, but there's no soft tissue. This can be beautiful type of materials. This particular sample is an agatized dinosaur bone, and they can be cut and formed very beautifully and preserve a lot of interesting details in them. But no soft tissue. If these were the only scenarios possible, then soft tissue would never be preserved. But nature includes a lot of different scenarios. And Center describes some alternatives, and more will likely be identified later as we go along. So in this scenario, our bone was laid down or is deposited in an oxidizing environment at the floor of a body of water. Okay, water microbes invade the blood vessels of the bone's cortex, that outer shell, and consume organic material. And as they do, they increase porosity of the bone, allowing more water to enter. Some decay is inevitable. The entry of water causes the collagen in the matrix of the bone's cortex to swell, and that creates cracks in the cortex that lets in even more water, accelerates the rate of collagen destruction, even after the initial microbial phase of decomposition has taken place. Now, if our bone was tiny, these processes would have consumed it before it had a chance to fossilize. Big is better. Dinosaurs have an advantage for big bones. And a bone like we're talking about here would be quite large, like from a T-Rex or a Triceratops. And it's very large, and so the main destructive processes are restricted to that external cortex, that last layer. As microbes in the blood vessels of the bone's exterior precipitate minerals that plug vessels, they block both themselves and water from entering further into the bone. Iron is released in its bone cells and in the lining as the blood vessels begin to decompose. The resulting Fenton reactions split numerous organic molecules and forge cross-links between the fragments, enabling those fragments to remain intact. Further Fenton reactions generate further breakdown of organic molecules, cross-linking between their fragments. This occurs as iron is released from decomposing blood cells and the release of the iron also generates a protective coating of iron oxyhydroxide over the internal and external surfaces of the blood vessel and bone, a coating that will be later converted into an iron oxide. Over time, crosslinks accumulate between the collagen molecules in the bone matrix that was shielded away from these Fenton reactions by bone material, increasing its stability. Over really long periods of time, water does eventually get into the formerly shielded blood vessels, but where the coatings of iron oxides over the blood vessels is continuous, the vessels are shielded from hydrolysis. When it's interrupted, the blood vessels succumb to decay as the water and other destructive agents arrive, leaving behind this empty shell of iron oxides that maintains their original shape. Some collagen also succumbs to the decay by hydrolysis and other destructive agents, but such decay is prevented where crosslinks are particularly abundant and where water has not entered into the adjacent vessels. Now, as groundwater seeps through, recrystallization and permineralization take place in parts of it, and that, again, helps to isolate it, helps to provide a means of preserving it. Dr. Center explains that by such means, in different magnitudes and modes of cellular and soft tissue preservation can exist in different parts of a single bone due to the differences in the microenvironments, the diagenetic history in different parts of the bone. Obviously, I've relied an awful lot on his analysis of this to describe this type of scenario. He studies demonstrate that each phase in this scenario occur. Such a scenario would preserve soft tissue for millions of years because it's chemically isolated or sequestered from the water and the microbes that bring decay. Such isolation would be effective until something breaks it down. Young Earth creationists need to explain what mechanisms they believe allowed dinosaur soft tissue to be preserved for 4,000 years. Then they need to show why mechanisms that allowed soft tissue to be preserved far longer than most soft tissue, why it cannot be extended over much longer periods of time. And I don't doubt that many young Earth creationists will continue to believe that soft tissue proves that dinosaur fossils are young. I don't think any particular scientific evidence that we might show would tend to sway them. But they should not claim that soft tissue demands a young Earth because there's increasing evidence of pathways that would be available, that would be realistic, that would preserve tissue for millions of years. Now, we're not going to be running any experiments for millions of years to show that. Would 1,000 years work? Would they like to suspend their judgment for 1,000 years? Is a young Earth creationist refusal to accept the scientists' proposals for how soft tissue was preserved based on the quality of the science, or the prejudice against the idea that things could be ancient? But what does all this show us about the flood geology model that most of the Earth's rocks were deposited a few thousand years ago by a single global catastrophic flood? What I see are clear indications that the beds where we find evidence of dinosaurs in were actually deposited over periods of time that are much longer than would fit their models. We saw this with dinosaur egg nests that were well ordered, that had eggs developed over several months before hatching, and they were in multiple beds stacked above one another. We saw this with dinosaur tracks around tree trunks and roots that grew in ancient swamplands. We saw this where dinosaurs walked around on land that had dried long enough for well-developed mud cracks to form. We saw that this occurred in many different levels in the same areas and in many areas all around the world. Now, some young Earth creationists claim that the discoveries of fragments of soft tissue in dinosaur bones proves that these cannot be millions of years old. But we've seen that paleontologists have discovered mechanisms that would allow soft tissue parts to be sequestered away from active decay processes. And if they remain sequestered, then there's no reason to say that there's a limit for how long such soft tissues would be preserved. Scientists were surprised to have soft tissue preserved, but this has provided an opportunity to learn a lot of new details about how fossils form. The discoveries call into question the young Earth creationist interpretation of Genesis, but they do not provide any reason to doubt that God created the earth and all of the life here. He created us, and He wants a relationship with us, and that is not contingent upon the age of the earth or the interpretation of Genesis flood. Thank you. I hope you've enjoyed these. I hope you'll join me again for other work that I do. Come and explore my webpage a little while. Thank you.